Welcome back, students. Today I will talk about evenness. First of all, we need to define what is evenness. Evenness is just the answer to the question how equally abundant are each of the species. A simple way to combine abundance and richness is just evenness. So, is there any homogeneity within the sample? This is rare that all the species are equally abundant, and so some of them are more abundant in general than the others. So the distribution of individuals among species is just the measure of evenness. So at the opposite two extremes of this evenness is just the dominance or rarity. Dominance, of course, is the number of species that dominate the ecosystem. A rarity is the very rare number of species in the place and in the area where we are sampling. There are some indexes to measure the absolute dominance, and one of this is just simple N1. So it's N1. So if N1 is the abundance of the most abundant species in the sample, there are relative abundance indexes also. One of these is N1 is divided by N, where N is the total abundance of the samples. Another measure is the Macnaughton's index of dominance, that is N1 plus N2 divided by 2N multiplied by 100, which means that N1 is the number of individuals of the most abundant species, N2 is the number of individuals of the second abundant species divided by the total number of species of individuals among all the species. The percentage index of singletons is another way to measure rarity, and in this case we just measure S1 divided by S, where S1 is the number of singletons that are species represented by only one individual, and S, that is the total number of species in the sample. If we want to measure the rarity, we have another way, that is PCT rare 1% index. This index is just the sum of S1 plus S2 plus etc. divided by S, where T is the biggest integer number less than 0.01 multiplied by N, that is the total abundance. We can use this only if the total number is more than 100. But if N is more than 20, we can use PCT rare 5%. In this case, T is the biggest integer number. The formula is the same as that of the previous one, but where T is the biggest integer number less than 0.05 multiplied by N. But if N is more than 20, we can use PCT rare 5%. In this case, T is the biggest integer number. The last index of rarity that we can use is the PCT rare N by S index, where on the exponent we use S1 plus S2 plus ST divided by S, where T is the biggest integer number less than N divided by S, where N is the total abundance and S is the total richness. A way to represent the dominance and rarity is the relative abundance graph. In this graph, we put on the axis the species number and on the ordinate the cumulative relative abundance. And what we can find in the curve, the inflection point. This point is the point that split into parts, species that are dominant and species that are rare. So we see that as much the line, the imaginary line that we can build on the inflection point is shifted to left, the less rare species we have in the sample. On this graph, that you see in the picture, this inflection point reaches almost 25 species. So we'll see that evenness increases diversity. In fact, when we have more evenness, we have greater diversity. This is true for all indices. For instance, in the example given in the picture, we see that we have the same number of species and the same number of individuals. But in the sample where the homogeneity and hence the evenness is higher, the diversity is also higher. The evenness can be used as an indicator of diversity, and for many ecosystems, high evenness is a sign of ecosystem health. It means that we don't have a single species that is dominant in the ecosystem. Often only invasive species are dominant, and this means that in the destructive site, dominance is very high because of a lot of invasive species. This is also a kind of paradox. The paradox of enrichment, where a polluted site, for instance, that are enriched by nutrients show a higher biodiversity. But in general, it is seen mostly in aged species. So it means that a simple biodiversity and with the dominance of a few ecological species, that means that is they are numerically abundance in dominance and in the number of individuals, but not in the number of species. It is important to understand that between ecosystem, comparability is usually not possible. It means that some areas have lower biodiversity naturally than others, and this is normal. For instance, tiger, that is not really much less even in the deciduous forest. Um, Seasonality also may confound the comparison as well. Earlier in those seasons where temperature is rising, for instance, it means that less even than later. So it's important to keep in mind that we cannot compare different ecological areas in this case. But if we want to have kind of comparison anyway, we need to try to prioritize areas for conservation that they are based largely on biodiversity. So not on ecological uniqueness. So evenness in this case could be a good representation, and we can use different ways to represent this. For instance, we can use a log 2 as the number of individuals in a graph in a histogram, while we put on the axis the number of individuals according to log 2, and on ordinate the number of species. Or at the same time, we can see that if we don't use the log 2 but we use just the number of individuals, the histogram will be shifted with the high abundant species on the left. This was noticed at the first time by Preston, who suggested the idea of veil line, where the veil line is just the line that hides the rare part of the species. So all the left to the left of this graph is completely hidden because of these rare species are not sampled. Useful representation of the evenness are the rank abundance plot or dominance diversity curve that are also called Whittaker plot. The rank abundance plot that will be explained later is just the species rank put in the axis and the relative abundance that is put in the ordinate. From the trend of the curve, we can understand the evenness of the species. In particular, if the central part of the curve was more or less sloped, we can understand if there is more or less homogeneity in the sample or not. So thank you for your attention. I will see you in the next lecture.